So let me go ahead and introduce our speaker today. Dr. Cynthia Aghogho is a plant breeder whose research bridges participatory evaluation, sensory science, and predictive breeding. She's currently a postdoc researcher at the Department of Crop, Soil, and Environmental Sciences at Auburn University in Alabama, USA. And today she'll be sharing with us insights on mapping consumer preferences of cassava using Tricot, which is a study that she conducted while she was at IITA in Nigeria.
Thank you very much, Rachel, for this opportunity and 1,000 farms. My name is Cynthia Aghogho, and I want to share a little bit of what we did in IITA in mapping consumers' preference using Tricot. So first of all, I would like to talk about why consumer-centric breeding matters, why we have to include consumers into the breeding program, especially in cassava. So when we talk about breeding in general, if you breed a material and it is not adopted by the farmers, it's like you're just wasting your time. And because that is adoption is where every variety is being judged. And a cassava variety will be adopted if it has the consumer preference, if consumers and processors are able to adopt it. And over time, we can see that the consumers have been prioritized lastly in the breeding program, which doesn't really give them the chance to test the varieties. So cassava, we know, is one of the major staple food in Africa. It is the second most important root crop, and 70% of cassava is being processed into gari, and from gari, most of it is turned into eba. And when we talk about eba, it is evaluated based on its quality traits, which is the color and the texture, which is like the moldability, the stickiness, and the firmness. And also in this study, we tried to map this color and textural traits and also map the overall acceptability of the product eba. So a little bit about the breeding stages in cassava. So breeding moves through several stages from the seedling to the on-farm trials. And at each stage, genotypes are being selected and diversity is being reduced. And because the consumer preference is being considered during the late stage, we might have lost some quality diversity maybe during the beginning of the stage. And the reason why consumer preference is being brought to the end of the breeding program is not because breeders don't want, but because there are challenges such as the processing time that is going to take, the stem perishability of cassava, and also the evaluation method of consumers' preference, which takes you to have to organize a panel, you have to organize surveys, which takes a lot of time, and logistics to plan, which the cassava stems do not have that time to stay on the field for you to do that. So that's why consumer preference is done during the on-farm trial stage, where we have little reduced number of varieties that can be tested and put out. So when we talk about consumer preference, it's mostly done using hedonic scaling. Hedonic scaling can be either a 5-point, 7-point, or a 9-point, which ranges from extremely like to dislike of a product. So you're giving the consumers a set of varieties and you're telling them to rate them either how do you like them, extremely like, or you like them, and you give them a score, which is either like a 5, a 7, or a 9. However, tricots, which is mainly used on-farm trials, they give you like just 3 varieties and you rank the samples from best to worst. So you're picking, okay, this is my best and this is my worst. So that's a difference between the two methods. One is your rating, while the other one you're just simply ranking 3 samples. So for hedonic scaling, you can get up to 7 varieties, 5 varieties. But for Tricot, you're getting just 3 combinations of varieties which are unique to a consumer. So bringing consumers into the breeding pipeline early and opens up opportunities to strengthen evaluation and accelerate varietal adoption. In this study, we evaluated both hedonic scale and Tricot side by side to understand which approach performs better in terms of the sample size requirements, like how many consumers do you really need, then the consistency in genotype ranking, and how each of this method connects to measurable qualities. So these measurable qualities that we evaluated in this study included the fresh fruit quality, gari and eba quality, things like the texture, color, viscosity of eba, starch property, flour property. So just things that we can quantify, traits we can quantify that we can later, maybe if they are connected to consumers' preference, we can maybe get genomic markers for them, or we can include them as proxy of selection so that we can simultaneously improve both consumers' preference and the agronomic traits that we select early in the breeding program. So this is going to actually help us shift from just breeding agronomy at early stage to also including consumer preference into our selection indexes using these quantifiable traits. So in this study, we used 19 cassava genotype, which included advanced breeding lines, the improved varieties, the land races, and they were all white flesh, and they had diverse dry matter content and fresh root yield, so that we'll be able to include enough variability and diversity into the study. So we took traits like biophysical, textural, functional, and color traits from the fresh fruit, the gari, and the eba. But on the consumer side, we evaluated the eba samples using both the classical and the hedonic ranking, like I said before. So the hedonics panel had 480 consumers, while the Tricot panel has 760 consumers. The testing, before each of these consumers were allowed to test, there were people that they consume eba regularly, and we had to get a diverse set of people from all age, ethnicity, background, working in IITA. Then we also evaluated 8 samples per section for the hedonic scaling, while for the Tricot, it's just 3 unique combinations given to one person. At the end of the survey, each of these 19 genotypes were tested 120 times for the entire panel. So finally, we analyzed the result to compare the consumer experience, the sample size efficiency, genotype ranking, preference quality, and also we try to do like a prediction performance using machine learning to know which of the traits are highly linked to consumer preference, so that we'll be able to see how to move that forward. So what we found, so first of all, before we could do any comparison whatsoever between these two studies, we needed to know that these consumers we are using, they are not different. So looking at the consumer characteristic, you could see that both the Tricot and the classical consumers, they are closer together. There is no differences between the population that was used. And again, looking at the geopolitical zones where these consumers came from, they came from almost across everywhere in the country. So we have a diverse representation of culture, and we also had diverse representation of both males and females in our panel, which is very good to show how people can distinguish the genotypes. So first of all, for consumers and enumerators experience, just to show how they enjoy both the Tricot and the classical survey. So we surveyed 76 consumers and we had 10 enumerators. So we used like an open-ended questionnaire to ask them what they should just freely describe what they like and what they found challenging in using both of this method in doing the consumer survey. So for the Tricot method, the most words that we got was few samples, fast, simple, and saves time. While for the classical, we had most dominant words were like many samples, the long scale, and it's time consuming. So first of all, we're already seeing that, okay, the Tricot method, it's kind of easier compared to the classical method, because in the Tricot method, you're getting just 3 samples, while in the classical, you're getting 8 samples. So we also try to see, okay, when we talk about Tricot, we had 760 people, while the classical, we have 480 people. Definitely, when you want to conduct a consumer survey, you want to use the least amount of people to give you the most effective result. Um, to get 760 people to do a survey for you, it's a lot. It's a lot, but so what we try to do is, okay, what is the minimum amount of people that we need to use to get the same results that we got from using the whole crew. So far we saw that Tricot, when it goes to like 40% of people in Tricot will be able to give you at least 90% correlation. Like the data you're going to get using 40% of people you use in Tricot, you'll be able to get that when you use 100%. While for the classical, you could get the same result, but there will be a lot of variability between the results that you're going to get. So for the genotype ranking, we could see that both the Tricot and the classical, they had like a correlation of 0.86 when it comes to how these consumers, they rank each of these genotypes. So you could see that there is an agreement between the Tricot method and the classical method when it comes to ranking. However, for like trait like modability, didn't really have much of a correlation between the classical and the Tricot, but at least 58% is still a lot. So we could agree that for genotype ranking, both methods, they effectively ranked the best genotypes. So when we talked about measurable traits, I mentioned earlier that we did like color, textural analysis, functional properties. So when it comes to breeding, we place very high importance in heritability, because if a trait is not heritable, it is difficult to improve those traits. So for this study, we try to put a threshold on traits that is going to be linked to consumer preference. So we put a threshold of 40%. So if a trait doesn't have enough heritability of 40%, it wasn't included in the next analysis. So far we did 58 we collected data from 58 measurable traits, and 21 of them had heritability less than 40. So for the next analysis we did, just 37 measurable traits was used. So looking at the correlation, comparing how strong either the Tricot or the classical is connected to consumer preference, we could see that both methods, they are pointing in the same direction, in the sense that if a measurable trait is negatively correlated with the consumer preference, both methods is saying yes, they are negatively correlated. But the only difference now is Tricot shows a stronger correlation, like it is stronger, and we had more significant correlation using the Tricot method compared to the classical method. So for all the traits that the classical method had significance, the Tricot method also had significance, but the Tricot method showed a stronger correlation or a stronger relationship. So we further went ahead to do some machine learning. So here we have 7 prediction models. So first of all, I don't want to focus on just the accuracy or the values right now, but first I want to focus on the type of model that performed best for each of this method. So because it tells us something about the importance of how consumer makes their decision. So we see that classical, which is a hedonic state, performs better with the linear models like the elastic net and lasso. This makes sense because with a 9-point scale, consumer experience their preference in small and gradual steps, which is like you're giving either a 1, a 2, a 3, a 4, a 5. So if there is a slight shift in the likeness of a particular genotype, they give it like a higher value, which is like maybe you just move from 5 to 6. So so the classical method just captures like a smooth and a proportional change in liking, which can help us optimize the quality among, probably you have your elite genotypes. You're already in your late stage, you have your elite genotypes, and once they are ready to be released, you can use the classical method to see which of them can rank better. But when we come to the Tricot, consumer rank only 3 samples at a time, and this usually reflects a threshold-based decision rather than a gradual one, which we saw in the classical method. And a product can be fine up to a point, but when it crosses a particular limit, it becomes not likable, which is what the Tricot is saying. For example, if we talk about the linear regressions, we are saying if the liking is going, if the liking is going, or if the color, for example, if you're increasing, okay, let me do the hardness. If the hardness is going up, the likeness is going up. But in the real fact, when it comes to consumers' preference, it doesn't work that way, because they'll get to a point where the color, the hardness will be too much, then the consumers are going to reject that particular varieties. So that is what the Tricot is saying. So the Tricot makes a better decision, and it is captured by the non-linear machine learning, which is like the LG-Boost and the random forest, and it shows a stronger performance under these models. So the classical can help us understand how much a variety is compared from one another, while the Tricot tells us whether a variety is to be accepted or not, which is exactly how adoption works in real time. So farmers, processors, consumers, they don't adopt based on tiny improvement. They adopt based on when a product crosses a particular threshold of satisfaction given to them. So that's why Tricot reveals like a make or break characteristics that determine whether these varieties will be successful in the real world. So further, we try to now which measurable trait is important. So we have seen how Tricot and classical performs, but now we want to see, okay, do the method, do they portray the same trait as being important? So by comparing the variable of importance from the top predictive models across both evaluations, we were able to identify that the trait that were repeatedly showed in both of the methods, they are the strong drivers of consumers' preference. So first, we found like a core predictors, which are traits that they were found in both methods, which is the traits like the starch residue, starch pasting property, hold back, and Gary said, but interestingly, there were traits like dry matter content, which is something that the breeders we already use in selection, and it has been shown to be strongly associated with Gary percent, which is like the yield of processing product, which has which has been something the breeders have been working on. So those traits, they showed up from just Tricot, which means, and if we go back to when I say make or break, Tricot is trying to tell us that if these traits and if these traits are not in the genotype, or if it's not there, there is a high chance that the genotype will be rejected. And dry matter was one of the traits that Tricot portrayed like it is important. However, dry matter alone does not guarantee good cooking quality, and it has been shown from studies being negatively correlated with traits like hardness measured using the textural profile analyzer. So while dry matter can help increase the economic value of a genotype, it is not a proxy for consumer preference, but it is important, as it's said. So what we learned so far from the study is that Tricot need with fewer consumers, you can also reach a reliable conclusion, which make it more scalable. You can do that at the beginning, at any stage of your breeding program. So both Tricot and classical agrees in the ranking of varieties and shows straight correlation signals, confirming that they detect the traits underlining the consumer preference in the same direction. However, Tricot connects more strongly with non-linear prediction models revealing threshold-based decisions than the classical method. And both methods highlighted some core quality traits which we can also look into further to see how we can improve them together with agronomic traits. And so together they provide like a complete understanding of consumer preference for use in breeding. So the Tricot and the classical method are not competing method. Instead, they are complementary tools that we can use, because the classical tells us how much a consumer likes a product, while the Tricot tells us whether they will be accepted in the real world when they are released or not. So this is the end of my presentation. Thank you very much for giving me the opportunity, 1,000 farms, and to the team, thank you so much.
Great. Thank you very much, Cynthia. That was really interesting and shows that the interpretation of consumer preferences is really not very straightforward, that you've had
Yeah.
to do a lot of comparisons on that. I've really enjoyed that.
Oh, thank you.
Lots of claps.
Oh,
Okay, great. Let's open the floor to any questions. Please raise your virtual hand if you would like to ask Cynthia a question. Okay, the first question is from Ondover Shau. Please unmute and ask your question.
Yeah, good evening in Nigeria. It was really a very good presentation, and I really enjoyed it. I wished one day we'll have opportunity also to present it on soybeans, because I'm a soybean breeder, and
Oh,
we need more of protein than carbohydrate. So one day we'll get to soybeans also. Thank you so much. I want to quickly ask, make input. When you said the strength of correlation, the tricot was stronger than the conventional. I also want to say, is there no possibility of partitioning the correlation to direct and indirect the regression? To know, because there should be other factors militating against the correlations. I don't know that my question is clear, because once you say the correlation is strong and it's positive, it's in one direction, is there no possibility of using part coefficient analysis to partition it to know,
to
is
see
it direct
how,
or indirect effect? I'm just trying to voice it out. Is there a
yeah.
possibility like that? Because since there are other factors also, there may be a particular factor that is giving that direction of the correlations. So other factors need to be correlated. If it is direct, the regression is direct, that means we can use that as a basis for selection. I was just thinking aloud. Thank you.
Okay. Oh, thank you very much, Shehu. I didn't really think about that, but we can do that and add it to our analysis. Thank you.
Great. Thank you very much. Next question. Let me just check the chat, see if there's anything. Just congratulations for the great presentation from several people. No
Thank you.
questions. Ismail, please go ahead and unmute.
Yeah. Good afternoon, good morning, everyone. So great presentation, Cynthia. We did this job together. But anyway, if I ask a question, you can consider it as a friendly fire. You said
Yeah.
in your presentation that breeders can use this approach earlier in the breeding selection. But you can imagine in this case 19 genotypes or 18 genotypes, and it took hundreds of participants to do the evaluation. So what is the earliest breeding stage that we can use this approach?
Well, I think the earliest breeding stage would be like in the AYTs when the genotypes are down. However, if there are traits that have marker data, or the trait that was correlated with the consumer's preference, we can add them into selection indexes to be like a proxy for improvement earlier.
Yeah. I mean, the whole idea was to use the best possible method for consumers to rank the genotypes, and then relate those rankings with the biochemical or biophysical traits to find those attributes that we can measure either in the lab or with markers or with the spectral data. Anyway,
Yeah.
well done.
Thank you.
Thank you very much, Ismail. Any other questions? Don't be shy. I've got one while we're waiting. Can you talk a little bit about the differences in costs and the duration of the trials?
Okay. For the cost, if we want to include giving out incentive to the consumers, we could definitely see that in Tricot you need more people, which is or we used more people, which is like 760 consumers. So definitely when it comes to giving out incentives, it's more expensive doing the Tricot. But when it comes to the duration, because a consumer is just spending like they spend more less time doing the Tricot compared to the classical, because you're getting just 3 varieties with the Tricot and you're just going to rank A, B, or C. But when it comes to the classical, you have to now think within a scale of 1 to 9, what are you giving, what value are you going to give a particular varieties? So before you think about 1, think about 2. By the time it gets to like the 6th variety, they're already having that fatigue. They're just, so when it comes to time, Tricot is a bit faster. But for the cost, if you want to consider giving incentives, which is not compulsory, it's the Tricot, it's more expensive.
And that's independent of the different sample size you had, because you had a lot more farmers involved in the Tricot than in the classical.
Yes, so it's because each of the consumer for the Tricot got a unique set of variety. No two consumer got the same combination of 3 varieties. But for the classical, 120 people got the same set of 8 varieties.
Okay,
Mhm.
thank you.
Yeah.
So I think Shahu had his hand raised, and then we'll go to Ugo and then to Edward. So
I think okay, Shahu. I thought
Shahu, do you have a new question or is that an old hand?
Yeah, no, it's a new question.
Okay, go ahead.
Okay.
What I want to quickly ask is, working with such large set of farmers, is there any enumeration given to them? Because once we are trying to do Tricot, now we discovered, okay, getting farmers together, they have been demanding for some stipends or enumeration and all these ones. I wanted to also learn from you, because there have been so much demand that you must make some commitment. That's one. The question number 2 is, when we come to a point of doing our feed day, brown feed day or demo, and since we have those smaller feeds in batches, not like the mother trial where you have a relatively bigger land or bigger area where you can do your farmers feed day. You bring farmers together, maybe 500 or 1,000 farmers in a place and do this. But the sizes of the Tricot, how do you manage with the on-farm? So, sorry, with the feed day. I can also learn from you. I wanted to learn from you so that I can apply it. Thank you, ma.
Okay. I am not involved in on-farm trials. What I do is consumer preference. So the consumer preference is the consumers that come to you. So we used a canteen where people come to eat and we give them the samples to test. So we did not go out to the farm. So yeah.
No, I'm saying because in soybeans, we plant on the field. So most of the preferences on the field, once we get to field, they may choose based on height, based on the color, based on the number, the pot size, and all these things. We get it right on the field there. Though your own is more or less coming back. There also, we need to do farmers' preference. So
Yeah.
when we have a
Yes.
smaller area, trying to say, okay, how do we manage the feed day? Because the people abroad were jam-packed in the whole place, over 200 or 300 persons. To
Oh,
have just a
wow.
small space. I'm just thinking so that I can also learn from you. Thank you.
Okay. So I think Bella, they are the ones that go for field day, Bella, or I'm not involved in field days.
Okay, yeah, I think Bella, I'll put Bella's email into the chat. If you want to ask him a question, Shahu, you can. Thank you. So let's go to Ugo, please unmute and ask your question.
Yes, thank you, Cynthia, for these great presentations. I have a question about your machine learning models. For me, it was not clear what are you predicting? Are you predicting rankings? Are you predicting scores or log words?
Okay, for the Tricot, we use the log worth, while for the classical, we use the rankings. So those are the predictors. So we use the biophysical, which are the measurable traits, to predict the log worth for Tricot, and we use that to predict the rankings for classical.
Okay, so in your inputs variables, you have your log words or your ranking data?
So log worth for Tricot.
Yeah. Okay. And then your predictors are the log words themselves. Or you have an a set of input variables and one output variable. So I'm curious to see which do you use for inputs and which one do you use for outputs?
So the inputs are the measurable traits. The outputs that we want to
Okay.
predict are the log words.
Okay. The observed traits. Okay, then it's clear. Okay, thank you. Thank you for the clarification, Cynthia.
Yeah, thank you.
Thanks, Hugo. Edward, go ahead and ask your question. Edward, are you still there?
Thanks. Yeah, I'm here.
Go ahead.
Took me a second to unmute.
It's all right.
Thank you for a really nice presentation. Congratulations. My question is a very simple question. I see that you've evaluated EBA and you've made sure that you got a good sample of people from across Nigeria. And I think maybe that there are other ways that gari is consumed. And that is like maybe in the form of, at least I'm in Ghana and they talk about soakings, and there may be other ways. And I just wonder if you have any indication about the way quality from one type of use might translate to another type of use across varieties, right? Are they specialized? Would you end up with a specialized gari for EBA and another for, more generally utilizable or something like that? If you have any ideas about that.
During the survey, one question that we asked is consumption pattern. How do you consume your gari? So, but most of them, they go for soaking, for EBA. However, when we talk about the quality of gari, which is like the biophysical trait, there is what we term as swelling index, which it's measuring how much a gari will swell when you add a particular amount of water to it. So from a previous study that we did, we found out that is also correlated with dry matter. So we did not in this survey talk about soaking, but at least we have an idea that the soaking quality of a gari can be correlated with dry matter. So we just added everything up.
Great. Thank you. Thanks.
Yeah.
Okay, super. Ismail, you have another question? Go
Yes,
ahead.
I have another question again. So in consumer ranking or scoring, when they presented a product, if there's a dominant trait they prefer and they score that trait high, there's a likelihood that they will also score the other traits high. And then there's this Tricot approach, which is to present trait by trait and say, do you like this one or you like the other one? Did you observe that, Cynthia? And and which approach most likely breaks this halo effect of when you like something, you score everything else very well for that product.
Yes, so I didn't present the result, but we have a very high correlation that if a particular variety is scored high for color, there is a high tendency that it's going to be scored high for every other trait, because you're giving them the varieties and they are ranking everything at the same time, compared to Tricot, where you give them the 3 varieties. Okay, which one do you like for color? Now, which one do you like for texture? So the Tricot breaks that ranking or that kind of decision making.
Okay. Thank you, Ismail, for your question. Any other questions from the floor or the chat? Nothing in the chat. Okay. All right. That's fantastic. I'll just share a couple of closing slides. Let's see. Sorry about that. I just wanted to bring to your attention, if you aren't already a member of our Community of Practice website, this is the kind of the homepage, and here you can introduce yourself. You can start conversations in the community section about Tricot and the work that you've been doing. You can ask questions. We even have some prompts and we ask you about things such as ranking versus scoring, and you can give your thoughts on that. And you can also see all of the previous webinar recordings on this. So it's a really nice place to interact. And it's very easy to sign up. You just go to the top right where the red circle is there and then sign up for free. And you'll get regular updates on the latest post from the website. So I just wanted to share that with you if you aren't. I know a lot of you are already members, but I really appreciate if you'd sign up. And with that, I will thank Cynthia for her wonderful presentation. It was really interesting, and I think a lot of people will agree with me on that. There are a lot of compliments in the chat, so thank you very much for sharing your work. And please come back again. And all the best to you in Auburn University in your postdoc. And thank you everybody for joining us as well.